Hello viewers, today we are going to discuss about a topic chromatography. It is a topic from biophysics. So, it is a common topic for various students who are studying BSc biological sciences, MSc life sciences, chemistry. So, chromatography is very useful technique, and in today's session we are going to discuss about various aspects of chromatography like principle of chromatography, instrumentation of chromatography, types of chromatography and applications of chromatography. So, all these we are going to discuss in today's session. Chromatography is a biochemical technique which is useful for separation of mixtures into their components. Once the components are separated from the mixture, the components can be analyzed, the components can be identified, the components can be purified and the components can be quantified. This means chromatography is not only useful for separation of components from the mixture, it is also useful for analysis of samples, identification of samples, purification of samples and quantification of samples. Coming to the name chromatography, initially this technique was used for separation of colored compounds. So, chroma means colored, graphy means to study. So, as it was used for separation of colored samples, it was named as chromatography. But now chromatography can also be used even for separation of colorless compounds like some of the amino acids. This technique of chromatography was introduced for the first time by it was introduced for the first time in the year 1906 by Michael Sweat, and this chromatography is a important technique in chemistry as well as in biology. So, the main use of chromatography is separation. Apart from separation, it is also useful for identification. So, every chromatography technique consists of various components. The first one is sample that is to be separated, second one is stationary phase and third one is mobile phase or moving phase. So, this chromatography is having number of applications. I will explain the applications of chromatography in the next video. So, coming to the definition, I just now I said chroma means color, graphy means to study. So, it is useful for separation of compounds. Initially it was used for separation of colored compounds and the name chromatography was given. So, every chromatography consists of three components: sample, second one is stationary phase, third one is mobile phase. So, based on the nature of stationary phase, chromatography has been classified into two types. If the stationary phase is a solid, it is called as a partition chromatography. If the stationary phase is a liquid, if the stationary phase is a solid, it is commonly called as absorption chromatography. If the stationary phase is a liquid, it is called as partition chromatography. If a stationary phase is a solid, it is called as adsorption, and adsorption chromatography is again of two types based on the nature of mobile phase. If the mobile phase is a liquid, it is called solid liquid chromatography. If the mobile phase is a gas, it is called as gas solid chromatography. Coming to partition chromatography, where the stationary phase is a liquid, is classified into two types based on the nature of mobile phase. If the mobile phase is a liquid, it is called as liquid liquid chromatography. If the mobile phase is a gas, it is called as gas liquid chromatography. So, paper chromatography comes under liquid liquid chromatography, whereas column chromatography comes under solid liquid chromatography, where the stationary phase will be solid and mobile phase will be liquid. So, you can see mixture is there. So, you can see yellow colored, red colored, blue colored. So, all are the mixture form. When subjected to chromatography with the help of stationary phase and mobile phase, you can see the yellow color components, red color components and blue color components are separated into individual components. So, this is the use of chromatography. So, mixture can be converted into components based on with the help of use of stationary phase as well as mobile phase. You can see how the molecules are traveling. Initially, the red, blue as well as green are at the same point. But once the chromatography is started, you can see the red is moving slowly, green is moving moderately, and blue is moving fastly, so that after separation, after some time, they will be separated as per their movement. So if I take the paper chromatography, okay, so the molecule having light weight, less weight will move fastly than the molecule having more weight. So based on their molecular weight, size, shape, they get separated from the mixtures, and the components can be get in a pure form. So just now I said the types of chromatography, adsorption and partition. Apart from that one, so there is other classifications based on the use as well as the nature of mobile phase, like liquid chromatography, gas chromatography, paper chromatography, thin layer chromatography, ion exchange. So all are there are various classifications of chromatographies are there. Coming to principles, so in today's session I will explain you in detail about paper chromatography. So paper chromatography is a type of partition chromatography. Why? Because here a solvent, that is liquid, acts as a mobile phase and a liquid acts as a stationary phase. As a mobile phase and stationary phase both are liquids, it is comes under partition chromatography. As we use paper as a supporting medium, this chromatography is commonly called as paper chromatography. This paper chromatography was introduced for the first time by Imzulo and Straber. And whatever the paper that we use in paper chromatography is a Wartman #1 grade filter paper. The reason behind the usage of Wartman paper is it contains only cellulose. It contains 100% cellulose, and the cellulose will be having high holding capacity of water. It holds water, and that water held by the cellulose molecule acts as a stationary phase. You can see the principle behind paper chromatography separation is capillary action, solubility. Okay, so capillary action and solubility, these two will play important role in separation of samples by paper chromatography. So as I said, every chromatography consists of three components: stationary phase, mobile phase and sample. So the paper chromatography experiment is very simple and can be done in a short period of time. So you can see the purpose of paper chromatography is separation of components from mixtures, and preparation time is very less. We can do the preparation in 10 minutes and the process can be completed within 45 minutes, and the setup can be done at the low cost. Coming to the materials, various chromatographies like paper chromatography requires various components. We can able to use chromatography chambers. If chromatography chambers are not available, we can able to use the beakers. We can able to, we need water, we need solvent. So different solvents are required for different samples, and we need Wartman #1 grade filter paper which acts as a supporting medium. We need a pencil, scale. So all these are the requirements. So first what we have to do is we have to prepare solvent mixture. For example, if you want to separate amino acids, the solvent mixture that we use is butanol, acetic acid, water. It is commonly called as bar system. Butanol, acetic acid, water. As per the composition, we have to prepare the solvent. Once the solvent is ready, this solvent is taken in chromatography chambers, and after preparing solvent mixture and pouring solvent mixture in the chromatography chambers, then you have to take Wartman filter paper. Okay, and you would cut the size of the paper as per the size of chromatography chamber. Then with the help of scale and pencil, a line is drawn from the bottom 1 centimeters above the bottom, and on the line the samples are placed in the form of spots. Then the paper is fixed in the chromatography chamber in such a way that the tip of the paper should dip in the solvent but not the sample. And after fixing the paper in the chromatography chamber, the setup is kept undisturbed for some time. Then what happens is, so because of adsorption and diffusion, the solvent will move on the paper to upwards, and when the solvent is moving in the upward direction along with the solvent, the sample also moves in the upward direction. And during this movement, based on the adsorption forces and molecular weight, different components will travel with different speeds. The components having more weight will travel slowly, and the components having less weight will travel fastly. So, after some time, the components will be at different places, and these components can be identified by observing the spots. If the components are colored, we can able to see the colored spots directly. If the components are not colored, we will convert them into colored form by adding a coloring reagent. You can see separation, how the separation is done. So, once the chromatography is over, we have to observe the spots. If the spots are visible, it will be okay. If the spots are not visible, we have to go for what? Coloring agent. For example, take colorless amino acids. The colorless amino acids need to be observed by making them colored by adding ninhydrin. Okay. So, separation, after separation, the spots can be observed. If they are colored, they can be seen directly. If they are not colored, we have to make them colored by adding coloring reagents like ninhydrin. You can see, observe in the chromatograms. So, initially 0% there is no separation movement. So, after some time movement started, you can see the blue colored, red colors are traveling, and after 50% of completion, you can see, so how the chromatography is there, 70%, 100%. So, you can see initially they are in mixture form, now they are separated. So, not only separation, with the help of the distance travelled by these solvents, these solutes can be find out, and with the help of these distances we can able to find the compounds also. So, there is a formula for calculation of RF. RF is equal to distance travelled by solute by distance travelled by solvent. Based upon the RF value, we can able to identify the unknown samples. So, I will explain you the identification other aspects in the next class. Meanwhile, you remember paper chromatography. In paper chromatography, paper is a supporting medium, liquid is a stationary phase, and a solvent, that is also a liquid, is a moving phase. So, hence it comes under partition chromatography. Paper chromatography is of two types: ascending paper chromatography, descending paper chromatography, and radial paper chromatography. And whatever the paper that we use in paper chromatography is Wattman #1 grade filter paper, which contains high amount of cellulose. You can see the photograph showing chromatograms of separation. You can see various components can be separated with the help of this. So, in coming classes, I will explain you about various aspects of this chromatography, like TLC. I will explain you TLC. I will explain you gel permeation chromatography. I will explain you HPLC. So, different versions of chromatographies and their uses will be explained in coming sessions. Thank you.